Thank you very much for this opportunity, and today I will report you some of the research we are pursuing in my lab, attempting to understand the biophysical function of CRISPR-Cas9 through computational methods. So I will give you an overview of the background and significance of this system, and then I will report about the biophysics and the biochemistry of genome editing and how we attempted to understand it through computational methods. And finally, there will be a chapter on the specificity of genome editing. I think that I don't have to introduce the importance of this system to this community. As you know, this system triggered a revolutionary phase in biology with applications in many fields. What we are interested in is in understanding how it works and how we can improve it. This system comes from a bacterial immune response against invading virus, which triggers the action of this protein Cas9 that binds RNA and uses its sequence information to target DNA and perform double-stranded DNA cleavages. As we like to say, this system works a little bit like an aerial silk artist, where there is a protein, that in this case is the artist, that first binds to RNA, changes conformation to grab DNA and match it. So there are several conformational changes, allosteric and off-target effects that involve the biophysics of this system. And we are interested in understanding how these phenomena happen through molecular simulations. Let's focus on the biophysics of genome editing. How does it bind nucleic acids? So, as you can imagine, these conformational changes that we are investigating are very large conformational changes of this protein. So, these are conformational changes that require long timescale dynamics, millisecond timescale, micro to millisecond timescales. And the confidence of our biomolecular simulations, I would say, is in this range. So, what we want to do is to get out of our comfort zone, to push the frontiers of our methods and reach scales that were not reached before. To do so, we use quantum mechanical methods, the classical force field, and of course, cryo-EM processing methods. And the first question we have been attempting to answer is the mechanism of RNA binding. So this was my first paper in which I investigated how the protein changes conformation to bind RNA. And as you can see, our simulations could retrace the conformational change, the very large conformational changes of this recognition lobe, and how it changes with respect to the other domain, to this arginine-rich helix, to grab RNA, and reconciling what was observed through the first cryostructures obtained through negative stain. Then we moved to the understanding of the conformational dynamics of the catalytic H&H domain. So this domain, the dynamics of this domain is important to regulate the function of Cas9, since by docking at the cleavage site, it performs cleavage, but it was also shown that the dynamics of H&H governs the entire function of Cas9. So what we have been doing is to map the conformational dynamics of H&H from the inactive to active state by running accelerated molecular dynamic simulations. When we started these simulations, the active structure was not present in the literature, and we actually found that by running extensive molecular simulations, the H&H domain would dock at the cleavage site dynamically in the microsecond timescale. So this observation was then was corroborated later, a couple of years later, by two structures appearing in the literature, two cryo-EM structures capturing the active state of H&H. And we found that there was a predicted error between the cryo-EM map and the atomic structure that we predicted, which was very low, which was actually a very good achievement for molecular dynamic simulations, I think. So based on this knowledge of the H&H dynamics, we decided to investigate how the H&H dynamics controls the allosteric function of Cas9. So two seminal papers appeared in the literature before us, two papers using molecular biology. In these papers, it was shown that the H&H conformational change governs the activation of Cas9, activating the catalytic function of the second catalytic domain, which is RABC. And this activation would be activated by the binding of a recognition region at a distal site, posing the basis for an allosteric control, which at that time was unknown. So to decide to investigate this allosteric control, we harnessed network models derived from graph theory. In particular, with these models, we treat every residue of the protein and of the nucleic acids as a node, and we calculate how these nodes correlate dynamically with each other and what are the preferred pathways between one node and the other. What are the pathways between, for example, one site that could be the H&H site and the RABC site? What are the shortest pathway, the dynamical pathway that maximize the correlation between one site and the other? So these are the same models that are used in Facebook and Twitter, where every friend is a node connected by edges. And of course, also as they do in social media, they can identify friends that are the key nodes of the network that are connected by more edges. And these are the friends where the information transfer passes much faster. You don't want to tell them your secrets, but this is basically the same concept. So we can identify key nodes and key residues of the system that govern the entire conformational dynamics of the protein. So we applied these methods and we identified what we believed to be the pathway for the information transfer between the recognition region, so where the DNA binds, to the nuclease domains. And what we could observe is that the pathway obtained with molecular simulations was overlapping with residues that were shown by NMR, by our collaborator, George Lisi at Brown University, residues that were shown to be slow through CPMG relaxation dispersion, which was an indication that this could have been the route for the information transfer, connecting the site of DNA binding to the site of DNA cleavage. So we then decided to, once we identified this pathway that you see here depicted in pink from the recognition to the nuclease robe, we decided to investigate what is the effect of point mutations in H&H that have been shown to improve the specificity of Cas9 against off-target effects. Are these mutations improving the specificity because they exert some effect on the allosteric pathway, on the signal transmission? So we used graph theory also in this case, and we built circular networks of communication gain and loss within H&H. So with these models, we could describe H&H as a set of communities of synchronized dynamics. Here, the A communities are the allosteric communities holding the majority of the slow residues identified with NMR that transfer the information from one side to the other. The other communities were not allosteric. So with the circular networks of communication gain and loss, we could identify if the communication between allosteric communities gets broken in the presence of the mutation, which is what actually occurs with an enhanced effect in one of the three mutations with respect to the other, which is this K855A. which was actually in qualitative agreement with what the experimental paper reported. The experimental paper reported is that the increased specificity of K855A was higher in the presence of a single point mutation with respect to the others. So this could already tell us that there is some relation between the way how this mutation interrupt the allosteric transmission with respect to the function, to the activity. So of course this was computed, and we wondered how this would have been real, right? So this is all what we wonder. And we decided to compare with NMR data, with the experimental dynamic exchange. So here you can see the H&H communities, which come from the computations that show synchronized dynamics. And in gray, you can we quantified the information transfer observed through NMR, which is the experimental dynamic exchange. And you can see that the tree mutation reduced the dynamic exchange in between the allosteric communities, which was supportive of what we observed computationally. So overall, these suggested that these enhanced specificity mutations perturb the allostery's mean H&H, and in turn, because of this reason, could impact the specificity. Of course, this is now an hypothesis that we are investigating in several other systems and has opened an entire field of studies. Okay, what about the biophysics, the biochemistry of genome editing? So Cas9 cleaves DNA, the two strands of DNA, through catalytic domains, H&H and RAV-C. Also in this case, we wondered, how is this happening? How does Cas9 cleave DNA? And to understand this mechanism, we use the quantum mechanical simulations where in particular mixed methods, where the core of the system is treated at a quantum mechanical level and the rest of the system is treated at the classical level. These the method that we use is based on molecular dynamics and is actually extremely expensive and quite complicated. It takes, for this simulation, takes a lot of time to run these simulations, and these are quite expensive. But it's actually very powerful. It is very powerful because, as you can see here in the case of RAV-C, we can follow dynamically the catalysis from the reactants to the products. So what we observed in our simulations is that an arginine residue that in the crystal structures was found to be in a different position was always pointing toward the schissyl phosphate, suggesting a stabilizing role. We also computed the chemical mechanism, revealing an associative mechanism that was then also in agreement with previous kinetic experiments, and was also in agreement with DNA cleavage experiments performed by Janice Chen at UC Berkeley. But what is most interesting is that when we started this project, the structure of the active site was not available. So magnesium were not introduced in the crystal and cryo-EM structures which were available before. And this arginine was actually in a totally different position. The new structure that appeared very recently shows that the active site that we predicted is actually very accurate. We then moved to the understanding of the H&H catalysis. In this case, two catalytic sites were proposed. One side, we called it pseudoactive, and this definition was given a posteriori. So this definition was given a posteriori because structures that were obtained in the absence of magnesium or by using experimental conditions that not completely resembles the real case scenario, suggested this active site in which this residue would point toward magnesium. But new crystal structures and cryo-EM structures proposed a different active site, an active site in which this asparagine would substitute the aspartate. So to understand which of the two structures is catalytic and how the catalysis occurs, we used a variety of computational methods. First of all, we performed free energy simulations to characterize the conformational change between the pseudoactive and active structure in the presence and absence of magnesium. And we found that with magnesium, the active structure was preferred, suggesting that previous structures supported this pseudo conformation because of the absence of the catalytic ion, and suggesting that the catalytic ion also hold a structural effect. Then we moved to the understanding of the chemistry. And in this case, we combined NMR experiments by George Lisi with our prediction of pKa, which was rigorously done through explicit solvent constant pH and replica exchange. And as you can see, the agreement in the case of the active conformation was actually excellent, suggesting that the active conformation would be the catalytic one. We then moved to the characterization of the catalysis through quantum mechanical simulations, and in this case we used a method called metadynamics that enabled us to move from the reactants to the product, showing that this is a histidine-activated mechanism, is an associative mechanism. And we also observed something very interesting, where this lysine engages in the protonation of the living group. So the way how we can look at this mechanism better is through a movie. You can see in the movie that the system is actually very large. These are more than 400,000 atoms. Is actually the biggest system ever studied with QMMM simulations. Here now we zoom into the active site, and then the catalysis starts. As I said, this is fully done at a dynamical level, so we can see how the system evolves from the reactants, where we are now, to the transition state, when we partially break and partially form bonds, to the products, up to the final observation of this lysine protonating through a proton shuttle through a water, the O3'. So this was an observation that required careful examination and experimental validation, which at the end supported this mechanism, because by substituting this lysine, the catalysis actually does not occur. Finally, I would like to spend some words on the mechanism of off-target effects. So off-target effects are the unselective cleavage of DNA strands that do not match the RNA guide, bearing base pair mismatches. So this work was initially inspired by single molecule experiment by Chen and Dagdas. In this single molecule experiment here, schematically depicted here, you can see that in the presence of 4 base pair mismatches at the end of the hybrid, H&H can assume an inactive conformation, unable to cleave the DNA. But in the presence of 3 to 1 mismatches, H&H progressively assumes an active conformation similar to what is observed in the on-target system, and is able to cleave the DNA. So the off-target occurs. And we wondered why this experimental observation. We wondered what is the molecular mechanism at the basis of this observation. So we did we used enhanced molecular dynamic simulations of the system in the presence of mismatches, and we observed that in the presence of 4 mismatches, there is an extended opening of the hybrid, which results in locking the catalytic H&H domain, in strong binding of the catalytic H&H domain, locking it into the inactive conformation, which could explain the reason why it does not activate, or is unlikely to activate in the presence of mismatch, of 4 base pair mismatches. So these results were very recently confirmed by crystal structures obtained by Martin Ginek, who has shown that 4 base pair mismatches at the end of the hybrid lead to this opening of the hybrid observed previously in our simulations. So based on this success, we decided to partner with Martin to investigate what would happen if the mismatches would be located along the hybrid. So first of all, we tested our method at positions where the crystal structures were available. For example, you can see in this case, and in this case, we can find a conformation of the adenine-adenine mismatch that is similar to what Martin observed. But for the other positions where crystal structures were, for which crystal structures were not present, we used enhanced molecular simulations to predict the conformations of mismatches. And this is a movie of the target interactions underlying the binding of mismatched sequences at the level of H&H. You can see the locking of H&H in the presence of 4 base pair mismatches. And finally, I would like to thank all of you for your attention. Of course, I would like to thank my group who has been instrumental in pursuing this research, in particular our graduate students, Akash Shaha, then the postdoc Pablo Arantes, Lukasz Niewcwicki, who did all the ab initio molecular dynamic simulations, and of course all the others, the funding agencies, our collaborators, and all of you for your attention.
So I was wondering, Ken Johnson's model for the catalytic turnover of CRISPR-Cas9 shows a single turnover model. Does your mechanistic simulations shed some light on that?
So the work of Ken Johnson, which is actually this one, right? So this is the work of Ken Johnson. So Ken Johnson reported the K-cut of the catalysis, and in terms of K-cut, we agree for both active sites, both H&H and RABC. What is interesting is that the K-cut in this case was measured after the binding of the nucleic acids. So 2 is a measure that basically is directly referred to the step of cleavage, which was great because we could fairly compare it with our simulations accordingly, of course, to the assumptions that are present in transition state theory. Regarding your questions of the single turnover, multiple turnover, our simulations looked at the step of cleavage, right? So we did not simulate what would happen after the cleavage. Which is something actually we could do, but I don't expect the system to give rise to very large conformational changes to enable a new cleavage, if this can answer your question. So maybe there is a point I could make to this audience that actually this is an experimental audience, and I think it's very interesting, this point. I think that we are assisting at some breakthrough by single molecule experiments, and it's actually very exciting to see that with single molecule experiment, you can measure the tau cleavage, which is the time required for actually the cleavage. So with single molecule experiments, you can measure the time required for the conformational change, then you can measure the time required for the cleavage. So I think that these can be parameters that can be used in the future to compare with our data. So this could be more accurate parameters to compare with our data, I think. Because we have many assumptions with this comparison. This is important to mention.
Okay, thank you, Giulia.